The thermophiles are actually aerobes and anaerobes both in nature. Okay, that one is basic difference from the methanogens and halophiles. Methanogens are anaerobic, halophiles are also anaerobes, but the thermoazirophiles are actually aerobes and anaerobes. They show both quality. Okay. They occur in hot sulfur springs. They are actually acid-loving bacteria. Under aerobic condition, these organisms oxidize sulfur to sulfuric acid and make environment highly acidic by lowering pH to 2. Under aerobic condition, what can they do actually? Under aerobic condition, they do their chemical machinery or chemical activity and convert that particular their surrounding environment into higher acidic nature. It means that approx 2 pH. And for that they actually oxidize sulfur. In form of sulfuric acid. So you have to remind this, or you have to keep it in your mind. That is under aerobic condition, the organism oxidize sulfur to sulfuric acid and make environment, or you can say to make environment highly acidic. Under aerobic condition, these organisms oxidize sulfur into sulfuric acid to make their environment highly acidic by lowering pH to 1 to 2. Okay, but what about under anaerobic condition? Under anaerobic condition, some of them can reduce sulfur. To hydrogen sulfide. Okay, look. And during anaerobic condition, they oxidize. Sulfur to sulfuric acid, while under anaerobic condition, they reduce sulfur to hydrogen sulfide. Okay, you have to memorize this. Okay, they are all also called acidophiles, or you can say acid-loving bacteria, or you can say oecinophiles. O is getting silent here. Okay, oecinophiles. So it is there are thermoazirophiles, halophiles, and methanogens. It's all about the Archebacteria. In this particular small section, you have to know about the nature of Archebacterians. That is anaerobic, aerobic plus anaerobic. Okay, you have to know, or you have to clear with the concept of their particular function. Like methanogen can produce methane, like halophiles is very capable to do light reaction for their metabolic machinery, or you can say they are very capable to produce ATP. And in case of thermoazirophiles, during under anaerobic and aerobic condition, they can do reduction of sulfur and oxidation of sulfur for their atmospheric condition or surrounding favorable condition. Okay. Now the next topic, or the next small lecture will be based on mycoplasma. What are the mycoplasma? Okay, look, mycoplasma are also known as monarians, or you can say cell wall less monarians, or you can say cell wall less bacteria. चलेगा, cell wall less bacteria भी इसको आप कहोगे फिर भी चलेगा. ठीक है. तो mycoplasma are actually smallest living body. It is very simplest kind of structure, and it occurs in free state always. हमेशा ये कैसा प्रेजेंट होता है बेटा? ये फ्री स्टेट में अपने को देखने को मिलेगा. फ्री लिविंग बॉडी होती है. ओके. नेक्स्ट वन, discovered by Nopod. It is discovered by Nopod and rocks. Okay. These two men are very important. Nopod and rocks discovered mycoplasma in pleural fluid of cattle, which is suffering from pleuropneumonia. Okay. Nopod and rocks observed or discovered mycoplasma in pleural fluid of cattle which is suffering from pneumonia disease. Their size very small. The size range is 0.1 to 0.15 neuron. Okay, one more thing, if in their condition the cell wall is absent, the main important point is cell wall is absent. So if there is no special structure, no special cell wall like hard covering, they show pleomorphism. They show pleomorphic condition. They can change their shape, or they can change their shape in different conditions. Or you can say they can show amoeboid nature in their shape. You can say, okay? Okay, envelope is present, but the main thing is that envelope is present. Okay, it means that cell wall is absent, but envelope is present. Okay, in case of genetic material, there is single DNA duplex is present. There is single means only one. DNA duplex बोले तो DNA double stranded DNA. Okay, so double stranded DNA की single chain यहाँ पे देखने को मिलेगी अपने को. The genetic material will be in naked condition. There is no any histone protein. Okay, naked condition. So histone is absent. Ribosome, there is 70 type of ribosomes occurs in mesosome, and for the replication, as the genetic material is initially not capable for the replication process. So for the replication process, a special disc-like structure present here. That one is called replication disc. That one are called replication disc. Okay. There in nutrition wise, they are heterotrophic in nature. Especially they show saprotropic and paratropic nature. They show saprotropic and parasitic nature. Okay, beta. Generally, they are disease-causing organisms. They cause disease in plants and animal both. For example, you can observe here in plants little leaf disease of brinjal, and witch brood. These two diseases you have to memorize. And another one in case of animal, pneumonia disease in domestic animal, pneumonia disease in domestic and animal, pneumonia, or you can say apleuropneumonia disease in other animals, and mycoplasmal urethritis in human being cells. So that is about some disease caused by mycoplasma. And you can look here, or you can observe here in case of diagram, there are no any special type of cell wall present, but the cell envelope easily can be observed here on the two layer. And in central zone you have to observe, or you can easily observe here the genetic, about the case of genetic material, which is approximately linear condition. Okay. From the initial position of the genetic material, there is a special structure known as replication disc is present here. Then genetic material, DNA and RNA, nucleic acid, both are present, but main genetic material is DNA. Okay, ribosome is 70 type, and the protein particles and granules and some enzymes are also can be observed here. So in this small section of the lecture, we can observe here about the respiration of bacteria, some primitive type of bacteria like Archebacteria, and some special type of bacteria called Mycoplasma. Mycoplasma will also known as pneumonia-like organism. Pneumonia-like organism, or you can say Mycoplasma-like organism, PBLOs or MLOs. Okay, so please don't get confused. Okay, you have to memorize what points you have to memorize about the facultative aerobic and facultative anaerobic condition. You have to memorize about the unique cell wall of the Archebacteria. You have to memorize about the methanogen, halophiles and acidophiles, respiratory nature, their habitat zone and their specialty. Okay, and in case of PPLO, you have to memorize the name of researcher, Bacterial rocks. It is very important. And another one, you have to memorize about the replication disc and the disease caused by at least two disease caused by mycoplasma in case of plant and animals. ठीक है, इतनी सारी सेशन के बाद अपना ये टॉपिक भी फिनिश. नेक्स्ट क्लास में अपन बेटा मिलेंगे साइनोबैक्टीरिया के बॉडी स्ट्रक्चर, उनकी बॉडी फिजियोलॉजी और उनके कुछ टाइप्स के बारे में। ठीक है, तब तक इसको रिवाइज करिए, इसको अच्छे से नोट्स बना लीजिए और स्टडी करते रहिए, खुद को हेल्दी रखिए।